So the genes can, or genes of synthetic DNA, and this has been manufactured, can also be made to help DNA synthesizing machines. Smaller chains of nucleotides, about 40 nucleotides, can be synthesized in this way. They can be linked together to make an entire gene or an entire genome. There are experimental implications for bioterrorism around the world. Campaign to eradicate polio, for example. Scientists have used poliovirus, widely known as genetic sequences, to synthesize that virus, the building blocks of DNA, in broths of chemicals. If these substances are produced, obviously, on a large scale by those who wish to induce harm, you can certainly see the implications of being able to use biotechnology, especially in the manufacturing of synthetic DNAs that can express the particular proteins for various types of organisms that can be harmful or even lethal in some cases. The results are not surprising but astounding in many ways. As your textbook refers, the DNA can be synthesized from the sequence, or it can be done by anyone that has the knowledge and some just basic equipment to be able to carry out this process. Poliovirus stores genetic information in long strands of RNA rather than DNA in a common laboratory machine that synthesizes DNA strands harboring the same proteins that are encoding the construction or instructions for particular DNA polioviruses can be carried out. So scientists are now more readily or more easily able to locate these long strands of DNA rather than RNA. And then in this genetic engineering process, these strands can then be inserted into plasmids, and then through a process of amplification can produce significant quantities of these organisms. One of the most common methods of selecting a desired gene is referred to as what we say is called blue-white screening from the color of the bacterial colonies that are formed in the end of the process. Only white colonies are of interest because they contain foreign DNA incorporated into a plasmid, and this will show up basically on a color screen. Further work would be needed then to identify the other specific gene sequences. So short segments, again, are single-stranded DNA consisting of a sequence of nucleotides unique to the gene sought would be synthesized. These molecules are called DNA probes. They're radioactively labeled, and so they can be located rather easily. The clone-carrying bacteria from the library, you use a library to be able to obtain your specimen and culture. They're grown into colonies. Plates on a specific type of nutrient agar is treated, and then it's open to the cells and it's separated the DNA into single strands. The labeled probe is then added to the plate. It'll react to a DNA colony. The colony base pairs will join. This is a colony hybridization. But now once the hybridization has taken place, remember you added a radioactive label to the other strand of the DNA. And so this will allow colonies to continue with the continued desired gene to be identified in the culture plate because you radioactively tagged it. It's not so unlike what we do in a clinical setting for people who undergo thallium scans. We actually radioactively tag specific red blood cells, and you're able to follow those throughout the system because you radioactively labeled them. Same thing here. If you're trying to follow a specific organism, you want to make sure you got an adequate number of copies, and you want to make sure the replication rate is where you want to be, and you want to be able to specifically identify the bacteria that you have now manipulated. You basically radioactively tag the parent DNA. You radioactively tag it, and then when you add it into a recombinant sequence, you're able to follow it because the radioactive base pairing will be labeled on the new organisms that are formed. So a similar probe based on labeled antibodies against protein products in cells also is used. The analogy I used for you was like what we use in thallium scans, which are particular types of scans that we use for people with heart disease. You tag the red blood cells, you can follow it through the system. You're able to follow the red blood cell because it has a radioactive tag on it. Same here, you're able to follow the daughter cells from the parent DNA because you tag the parent DNA with a radioactive tag. And this just goes through the sequence of genetic engineering again. Recombinant plasmids introduced into bacterium that will become penicillin resistant. You could also tag these radioactively, and so when you put the treated bacteria into a spread nutrient agar plate, this one contains ampicillin. You're trying to identify which ones are now ampicillin resistant. The white colonies reappear. They must contain the foreign DNA. The blue colonies must not contain the foreign DNA. It's just a different method to be able to identify particular types of organisms. Now, E. coli, you may have seen E. coli was used on a lot of these recombinant DNA types of procedures to produce either pharmaceutical products. We talked about erythropoietin, which is a drug that's used in patients that are anemic. Factor VIII, which is used in patients that have bleeding disorders. Insulin. E. coli is an organism that's commonly used in that. I know we studied E. coli the last portion of the lecture, particularly about some of the diseases that it caused, but there's also some positive implications as well. Remember, only a small percentage of the bacteria that are out in the environment cause disease. The overwhelming majority of microorganisms do not cause human disease, and many of these same microorganisms in the right setting don't cause disease, but even can help us to find medical cures and treatments. E. coli, used because it easily is grown. The genomics of it are well known, and so in this process, though, because we know E. coli can cause disease and can cause problems, we need to eliminate some of the toxic effects of them. E. coli has a powerful endotoxin that not only causes problems locally, so if you get an E. coli infection, say in a localized area of your stomach, an E. coli infection in the bladder, it secretes a toxin. The toxin gets in the bloodstream and can pass to other parts of the body as well. So because it has an endotoxin, we have to eliminate that. So the cells must be lysed to get a particular product. E. coli also is often used in genetically engineered organisms to produce desired gene products. Has disadvantages, though. That's the endotoxin. Endotoxin causes fever, can even cause shock. Shock is a condition where your blood vessels dilate. You have a particular blood pressure. Blood pressure, say, is 120/80. But if I dilate your blood pressure, your blood vessels, and open your blood vessels up, your blood pressure will fall extremely low. That's life-threatening. Your blood pressure has to be maintained at a certain level. So the endotoxin that's secreted by E. coli causes shock, causes your blood vessels to dilate, causes your heart rate to go up, causes you to have severe fever. These are the symptoms of shock, and all caused from the endotoxin. So it does secrete the product. The cells must be harvested, ruptured, and products must be recovered. So it has to go through this process of splicing out portions of the gene that are undesirable. The production of endotoxin would be one of those undesirables. Organisms such as gram-positive bacterium, such as bacillus. Now, E. coli, remember, is gram-negative. Gram-positive bacterium, such as bacillus and yeast, more likely to secrete their products. Animal viruses such as vacciноvirus have been genetically engineered to produce vaccines. Then mammalian cells and plant cells are often engineered to produce useful products as well. Medically important products are made by genetic engineering include, should be somewhat familiar with these, insulin, growth hormone, TPA or tissue plasminogen activator. This is a substance that's used a lot in emergency room settings and cardiac labs. TPA is a substance that eats up clots. If you get a blood clot, for example, you get a blood clot in your leg, blood clot breaks off, transfers to your heart, goes through your heart, goes into your lung. Now, those of you in the A&P2 know the clot comes into the right atrium of the heart, goes into the right ventricle of the heart, then is pushed out the right ventricle of the heart through the pulmonary artery, gets into the lungs where it causes major problems. Now, TPA is a medicine that will dissolve the clot. So if a person has a pulmonary embolism or a blood clot, you give them TPA, the TPA will dissolve the clot. Its main application is used in patients that have heart attacks. Patients that have heart attacks typically have blood clots that block the artery. Now, the artery has a certain size. Let's say we ate a lot of hamburgers and beef and all that stuff, and now the size of the artery is narrow. It's gotten much smaller. The lumen is small. Blood vessel is still the same size, but the lumen is small because you got plaque built up inside. Now, all of us pass these little tiny blood clots every day through our coronary arteries, but our body chews them up and it doesn't cause problems, and we got nice big lumens. But if the lumen narrows, these little blood clots now get plugged up. They block coronary arteries. They block the coronary artery. Heart tissue dies. When heart tissue dies, it doesn't regenerate, right? Cardiac cells don't go through mitosis, correct? Doesn't go through mitosis because cardiac cells don't have the T word. Centrioles. They don't have centrioles. So cells without centrioles don't go through mitosis. So if you have a heart attack, that tissue's dead. But with TPA, if you give it fast enough, it dissolves the clot, opens up the blood vessel, doesn't make the lumen big anymore, still a little tiny narrow lumen, but at least blood can get through now. So it's a very important drug to be able to utilize, but you need to use genetic engineering to make enough of it because we can make tissue plasminogen activator. And then vaccines, we already talked about flu vaccines and stuff that are produced, again, genetic applications, genetic engineering. So everything we talked about today brings you to this point. What's the applications that we see in a clinical setting? Medically important products. Medically important products include insulin, somatotrophin, growth hormone, tissue plasminogen activator, subunits of vaccines. We already talked about insulin and all those other things. Applications of genetic engineering, 1982. Food and Drug Administration approved the first time medical use of recombinant DNA protein for the hormone insulin, which was cloned in large quantities, inserting the human insulin gene, the gene responsible for the production of insulin in the human body, the makeup into E. coli bacteria. Previously, this hormone was used by insulin-dependent people with diabetes. Now it's available. It was originally only available in limited quantities from hogs, but a lot of people had pork allergies. There were a lot of religious concerns about using pork as a product as well, and so many individuals couldn't take advantage to insulin that was produced utilizing pigs. Now, through genetic engineering, we can make mass quantities of E. coli. Sensitivity levels to insulin now are basically or close to zero, so it's a much more efficacious product. Also, 1982, Food and Drug Administration approved genetically engineered protein for humans. 3 cloned hamster cells cultures produce tissue plasminogen activator, or TPA, enzyme used to dissolve blood clots, again, also used to produce erythropoietin, which is a substance that was only produced originally in the kidneys, and now they can produce that as well utilizing E. coli. Also hormones used to help people with severe anemia, anti-hemophilic factor 8, also used now and produced by using genetic engineering. So some very important applications. Another important genetically engineered drug is interferon. I already talked about that earlier. Interferon helps to fight viral diseases and very important as an anti-cancer drug. Recombinant DNA also utilized in production of vaccines and diseases. Vaccines contain portions of infectious organisms that does not cause severe disease, but it does cause the body's immune system to form protective antibodies against organisms. When persons are basically vaccinated against viral diseases, the production of antibodies is actually a reaction to surface proteins that coat the virus. When recombinant DNA technology, scientists have been able to transfer genes with some viral coats to proteins to the cowpox virus, which was used against smallpox first in the 18th century, and we talked about that historically. And then vaccinations with genetically altered smallpox is now being used against hepatitis, influenza, and the herpes virus as well. The most elegant method of introducing recombinant DNA into a plant cell is called the Ch09b plasmid. It's a bacterium that infects Plants normally carries the plasmid. The infection causes a tumor-like growth called the crown gall. And basically, the plasmid also serves as a vehicle for insertion of genetic engineering DNA in plants. Not really a big application for us, and I don't spend a lot of time on that. It's just a little bit of trivia that you may or may not be interested in. I'm going to assume by your facial expressions you're not really interested in that. It's okay. Scientists have also developed ways of using this, especially in agribusiness. I skipped over the zucchini. But in agribusiness and agriculture, recombinant DNAs have been used to combat some of the major problems in food production, destruction of certain crops, plant viruses by insects and pests. By transferring the protein code of a gene of the zucchini yellow mosaic virus to squash plants that had previously sustained damage from the virus, scientists now are able to create a transgenic type of squash plant with immunity to certain viruses and stuff. So that's very important to certain applications in agribusiness. Scientists also have developed certain types of potatoes and strawberries. Strawberries are important for us in our area that are frost resistant. Imagine the implications of this worldwide. Though our growth is well under control in the United States, I say that loosely, but it basically is. Our population is pretty stable, and then our country, other than with oil, we have not exceeded our resources. But in some countries, for example, like China, where we have 300 million people, they have like 4 billion people, and so food is going to be an issue. Genetically engineering certain types of crops that are resistant to different types of viruses that can damage the food industry in those countries. Genetically engineering particular types of fish and salmon, which are grown in farms now, have widespread implications of recombinant DNA and being able to use this in agribusiness as well. So it's very important. Being able to develop substances, chemical substances that will specifically kill the weeds that grow in farms and not affect humans, such as the herbicides and pesticides that were used many, many years ago. Scientists have also transferred growth hormone genes into rainbow trout, like I was alluding to earlier, directly into the carp eggs. Now you have these super carp, super fish. Probably saw it on Science Channel. They go around and eat people. No, I'm just kidding. But the resultant transgenic carp produce both carp and rainbow trout with growth hormone. So now we got bigger fish that reproduce much more faster. May not sound like a good thing where we are, but when you think about countries that have already fished out their seas or already fished out the ocean, fished out their local lakes and local ponds, you have a genetically engineered fish that's going to grow bigger and faster. Some definitely advantages to that. Now, if you're an environmentalist, if you're an environmentalist activist, even worse, but if you're an environmentalist, there are some concerns about this type of genetic engineering. How does it affect the environment long term? If we can get outside of our lifetime when we look at the whole picture, maybe, maybe not. Maybe it is a good thing and then maybe not. But certainly utilizing this technology enables countries to feed large populations of people where before that was not available. Bovine somatotrophin, basically it's used to clone large quantities of genes responsible for cattle growth. We already see that application now, not only in cattle, but we also see it in chicken as well. And we use, again, a bacterium, E. coli, utilizing recombinant DNA to be able to produce this. The hormone is then extracted from the bacterium that is produced. It's injected into dairy cows, increasing milk production 10 to 15%. Now, of course, they haven't been able to confirm all the scientific evidence for this, but then that hormone is ingested by you when you drink milk. Maybe, maybe not. Maybe it causes long-term effects, maybe not. But certainly there are some very positive effects to this. There may be some negative consequences as well, so just keep that in mind. Again, agri-application, bacterium, Bacillus thuringiensis is basically engineered into plants to produce a toxin that kills certain plant pathogens that feed on the plant. Again, genetically engineered product, bovine growth hormone, increases milk production in dairy cows. But going back to the bacillus here used, again, looking at this application. Previously, we worried about pesticides in the environment. Now you don't have to, well, we still do, but if we became a more agri-friendly business that utilized genetically engineered pathogens specifically for the pests that may affect the growth of crops, then at least those herbicides would not be exposed into the environment. And then therapeutic applications, subunits of vaccines, non-pathogenic viruses carrying genes for pathogens, antigens as vaccines, gene therapy to replace defective or missing genes. The Human Genome Project was a big project that was undertaken by a number of different countries many years ago to be able to identify the nucleotide sequences for the human system. Recombinant DNA technology was also used in the basis of DNA analysis of genetic abnormalities responsible for various diseases. It also contributed to advances in gene therapy, which abnormal genes might be replaced by normal genes in living individuals. There are techniques utilized, the southern blotting technique, which used to isolate fragments of DNA contained in a gene. This method, specifically referred to as southern blotting, you should know that, at least that definition. That will most certainly be on your test. DNA fragments from each candidate clone to be treated with the same restriction enzyme, and the resulting fragments separated by what's referred to as gel electrophoresis. Different proteins have different weights, and in gel electrophoresis, these different proteins separate out by their different weight and is able to identify particular fragments. The fragments are called restriction fragments, polymorphism, and can be identified utilizing various methods. Southern blotting, the bands represents fragments that are blotted out at a special filter, which are bathed basically radioactively with tagged probes. We talked about radioactively tagging particular components, DNA sequences previously. DNA complementary to a specific gene would be desired. The desired band can then be cut out of the gel sequence and recovered by soaking it in a particular solvent. And the genes can be studied by DNA sequence to determine the specific nucleotide that's there. Another way to be able to determine the sequences is using what's called a random shotgun sequencing. I like that name, random shotgun sequencing, of small pieces of genome that are sequenced when they're assembled in a computer analysis. The techniques are basically vital for identifying the human, when used in the Human Genome Project, shotgun sequencing. DNA sequencing, which is often highly automated, combined with the PCR technology that we talked about before, polymerase chain reaction, permits the recovery of detectable and identifiable DNA, and then it can be also used to identify extremely small samples. These procedures were used, so-called DNA fingerprinting, where each individual has a very specific, identifiable DNA sequence. If you can get samples of DNA from a given individual, it gives you a 99.9% accuracy in identifying that particular individual. This is used in the analysis in crime scenes. If you can get a sample, a DNA sample from a victim or a person who has created a crime, we'll be able to specifically identify, connect each the person that committed the crime to the person that was victimized at the crime just by obtaining a small DNA sample. So it's extremely important. Some of the other ways you can see it applied is in paternity testing. In paternity testing, obviously getting a sample of blood from the parent, a sample of blood from the child helps to identify. In violent crimes, if we can get just the hair follicle from the individual. Many victims that are victimized at crime scenes, one of the things we do at a crime scene, and we do in an emergency room, in the case of assault or rape, we take brushings from the victim, and we collect those brushings basically on a white sheet of paper that has no residue or debris on them. And then that whatever scrapings we get from that person, we analyze it in a microscope. We look for DNA traces from individuals. Now, we don't see that under a microscope. It has to go through chemical testing with CSI. But basically, you look for any DNA fragment that doesn't match up to that individual. A hair, a piece of skin, a nail, whatever, anything that would contain information about the attacker's DNA. And then as you get victims, you probably will see this on Law and Order or CSI all the time, they ask for DNA. Just one sample of your blood. If you're innocent, give me a sample of your blood. If you're innocent, give me a sample of your hair. And you can match that DNA with residue that's found from the victim. That's why at a crime scene, the clothes of the victim, the carpet of the victim, whatever material was at that crime scene is collected, because if you can find anything that contains DNA from the person who committed the crime, whether it was a violent crime, assault, whatever, robbery, if you can find any piece of DNA, you can connect that up. Now, the big question, the big controversy that's out there now, can we keep DNA libraries of known felons, people who commit crimes? Can we collect the DNA? Oh, better yet, when you're born, can we make a DNA library? We get a little bit of your blood anyway, so now we create a DNA library, and so if you're accused of a crime, boom, or if they find DNA at a scene, there's a library, just as they have for fingerprints right now. You use that library and you'll be able to bring up the genetic information of every individual. I don't know where you're at on that or not, but the technology is there to be able to apply it, and it's only going to get better. Not a bad tool to be able to utilize, especially if you're trying to identify people that do bad things. The human genome, is it a violation of privacy? Maybe. The human genome, goal of the Human Genome Project was to map out the genes in the human genome, 35,000 to 70,000 genes, approximately 3 billion nucleotide pairs. The next goal is the Human Genome Project, the Human Proteome Project, which will map out the proteins that are expressed by human cells. Completed in 2003. It's a 13-year project. It was taken over by the U.S. Department of Health and Energy and National Institutes. There were a number of international companies that attempted to start it off first, but because of funding or other projects, the United States kind of took the lead in it. During the early years of the project, the United Kingdom became a major partner. Also, some additional contributions from Japan, France, Germany, China, and others were able to participate in this process. So again, under DNA, sequence of organisms of genome, DNA fingerprints, this is an example of utilizing the isolate of patients whose infections were not related. E. coli isolates from patients who drank contaminated juice and apple juices. Again, you see particular enzyme products that are produced in each of the individuals and able to match up. You match up and you're able to identify the similarities between organisms, particular type of E. coli that were in people that drank contaminated juice. This would be E. coli from people who contained or perhaps had E. coli, but it was not related to that was in the juice. So you can see from this sequencing here that you can easily identify different types of E. coli, a strain that did not cause problems, a strain that did cause problems, being able to match up the identity based upon the specific sequences of the genome of that organism. Take that same application, so if you wanted to match up a mother and a baby or a father and a baby, you look at the similarities. If this would be the genome or the base sequencing of the baby, then the parent should match up pretty closely to the same. And that's just the basic principle by how this is used in applications. Again, clinical applications, crime lab applications when you're using DNA fingerprinting to be able to identify victims with attackers. This just goes over, you can go over this on your own if you want to go through the process of southern blotting. I do want you to appreciate the definition and its applications of it, but you don't have to know each of the steps that are involved in being able to do that. Now, and just hit one point on the ethics of genetic engineering. Laboratories obviously are engaged in recombinant DNA research. There are rigorous safety applications, accidental releasing of genetically engineered microbes. I talked about the genetically engineered virus that was passed out to different labs throughout the world. This was last year. It got a lot of negative press because it was a very virulent strain of a particular type of virus. The lab thought they were doing the appropriate thing because they were trying to test the quality of the labs to make sure they could identify different viruses, and they could. However, the strain that was used brought out a lot of concern because it was so virulent, and if it had escaped, what the problems could have potentially been. But microbes may also be used to engineer and contain what's a suicide gene that it would prevent them from surviving outside of a specific environment in a particular lab. So in other words, if you wanted to do study on smallpox, you could genetically engineer smallpox that it would have to be in a particular lab utilizing a specific nutrient at a specific temperature. So if it's stolen from that lab and it's taken to another place to try to mass produce, unless those individuals knew what the parameters were for that particular now new recombinant type of smallpox virus was, then the virus would die and you couldn't cause disease from it. That's also the case in producing some, mass producing some microorganisms. You may be able to manipulate the genetic components of the base sequence of it so it's not virulent anymore. It may have an endotoxin. Say E. coli, for example, has a particular endotoxin. Well, if we know the gene sequence that's involved in producing the endotoxin, remove that base sequence or put in an analog so you'll still get in E. coli, but it won't produce the endotoxin that causes problems. So there are ways that you can manipulate or engineer particular types of organisms so they will not survive or they won't cause virulence outside laboratories. There's also genetic screening that's utilized for hereditary diseases of birth, defects in the fetus that introduces a number of ethical questions. Do you induce a suicide gene that if a baby has a particular genetic disorder that it won't survive? Do you introduce or do you identify based upon genetic information or DNA sequencing if a baby will have a birth defect and whether or not you decide to carry on the fetus throughout the complete gestation? Do we screen parents based upon genetic information? Can you pre-screen the parents that have sickle cell so two parents that have sickle cell don't marry and don't have an offspring that has sickle cell? The applications can kind of go on and on and on. I'm not saying right or wrong, but it's just things that I want you to think about when we talk about genetic engineering, the possibilities and what we're able to do. In fact, able to even genetically engineer an offspring, a human offspring, without disease, without medical problems, or the potential for developing those problems because you're able to manipulate the genome. A lot of safety issues. Avoid accidental release of particular types of organisms, as I've already alluded to. Genetically modified crops for safe consumption of the environment, which probably is a good thing, but still is laden with some kind of some debate. Who will have access to an individual's